And now we're going to do is just go and look at our warm-up, which we typically do here. So it's going to just take me just a moment to get into this. And here we go. I just need to share screen. And here we go. All right, so warm up. All right, concentration. So this is a big thing that's going to be coming up for today and for, again, the rest of the semester, because ions crossing membranes are going to be a key way voltage changes happen and cells signal each other. And which way they go depends to a large degree on the concentration gradients. Obviously also depends on the voltage, the electrical force, but the diffusion gradient is super important. So it's important to know which ion is more concentrated inside versus outside the cell. You know, for your exam on Tuesday, I promise you there'll be questions that have to do with making sure you know which is more concentrated inside versus outside. Sodium and potassium in particular, we're going to be seeing a lot of. We'll see much more detail today. Again, we talked about there being that little sodium-potassium pump that's always running. So sodium, is sodium inside compared to the outside? What is sodium like inside compared to the outside?
Lower.
Much lower. Sodium is like only 1/10 as much inside as outside. So sodium wants to come in due to its diffusion gradient, as well as we're going to see due to the electrical force, because a resting cell is negative inside. So that's also going to want to pull the sodium in. So if you open a sodium channel on a cell, the sodium wants to come in. And as the sodium comes in, which is a cation, you're going to start having the voltage go up. You're going to depolarize the membrane. So that's going to be something to remember. Sodium channels open up, that's going to cause depolarization, the voltage starting to rise on the cell. Potassium, on the other hand, is going to be much more concentrated inside the cell. Again, 30 times as high concentration inside as outside, because you're pumping potassium in constantly. Again, so that's why when you open up the potassium channels, potassium wants to leave due to its diffusion gradient, which creates a negative membrane potential, which is what we find in the resting potential, because a cell is mainly permeable to potassium. So since the potassium is leaving due to its diffusion gradient, you end up having a resting potential that is negative. Again, a typical resting negative potential is about -70 millivolts. If it was pure potassium permeability, we'd be all the way down to about -90 millivolts. Chloride is another one important to keep track of. Chloride is much more concentrated outside the cell. So when we open up chloride channels, they are going to want to come in, even if, you know, because typically if we have a negative potential on the cell, you know, chloride doesn't want to come in towards the more negative inside, but because of the diffusion gradient, the chloride does come in. So when you open up chloride channels, what's that going to do to the membrane potential?
Make it more positive.
More negative.
You make it more negative, because the negative is going to come into the inside of the cell. That's going to make the cell more negative inside. So that is going to bring the voltage down. We're going to see that in just a little bit when we talk about inhibitory postsynaptic potentials. If you open up a chloride channel, you're actually making the neuron less excitable by letting the chloride in and making the overall voltage even more negative. Continue.
Professor, question about that. Is that something, because I know the sodium and the potassium we went over, but was this something like you can intuit that, like how it's going to be in terms of the, if
No,
it's low or higher, or that's something you have to,
you need to know. I mean,
okay.
it has to do with the balance between the diffusion force and the electrical force. So you just have to accept on faith right now for me that you open up a chloride channel and the chloride's going to come in and lower the voltage.
Got it. Thank you.
What is the voltage rest for chloride ion?
I'm not even exactly sure, and it's not so relevant because you never have the potential, the membrane being primarily chloride, so it's not as relevant. We're going to be flipping back and forth between major potassium, major sodium, so there it makes a difference. It's important for you to know the resting potential, or the equilibrium potentials there. But for chloride, it's not going to play a role like that. It's going to be more just for these small alterations of the membrane potential. Cytochromes, the cytochromes that make up the electron transport chain. So here we got a lot of people, did get this right, but a lot of people got it wrong. You definitely want to understand the different steps that are going on in cellular respiration here. They are definitely not responsible for the production of NADH. In fact, they are accepting the electrons from NADH, which is breaking apart in order to have the electrons run through to pump the protons. Where is NADH actually getting made?
The Krebs cycle.
That's happening in the Krebs cycle and a little bit in glycolysis as well.
Okay.
So, and then that NADH is contributing the electrons that run through the electron transport chain. Where do we actually assemble the ADP and phosphate to create the ATP?
Electron transport chain.
No, the electron transport chain does not do that. That's why it was wrong.
ATP synthesis.
Yeah, that ATP synthase. So it's that other membrane protein that is using the flow of protons back into the matrix of the mitochondrion that drives the actual manufacture of the ATP. So the ATP is manufactured through that ATP synthase that is then using that proton gradient, which was set up by the electron transport system. So
We don't.
question?
I'm sorry. So we don't consider the ATP synthase to be part of the electron transport chain?
No, not at all. It's a separate membrane protein that is taking advantage of the proton gradient that is set up by the electron transport chain.
Got it. Sorry. I've always learned it as being part of it, basically, and just like the end of it. So that's why I...
No, the end of the electron transport chain is just handing off the electrons back to oxygen to make water. And in that process, you're making or creating and maintaining that proton gradient. And then that proton gradient is now having the energy, and then that is then used to drive that ATP synthase protein to actually manufacture the ATP. So, they're all part of the same overall process, but they're fundamentally separate pieces if you start getting more granular, which we are in this class. All right. How many ATP are made by glycolysis? Here, most people got this. Only 2 net ATP. And again, for some things that we look at, that's going to be enough. If you want to get the full 36, you got to go into the mitochondria and use oxygen and go through that whole rigmarole of Krebs cycle and electron transport and ATP synthase, and then you get the 36. But just from glycolysis, which is happening in the cytosol. Without oxygen, you're only getting 2 net ATP for every sugar you start with. So again, that's going to be important to keep track of, particularly when we look at muscle metabolism a little later. Which of the followings is not an intermediate in the metabolic pathways involved? So this one here, I put this in here partly also just to make sure you remember the definition of intermediate. Again, what is an intermediate in a metabolic pathway?
Something that's made and then used as like a reactant or a substrate for the next reaction.
Exactly. So it's a compound. It's one of the compounds that's made along the way. And all of these are compounds that are made along the way in, first pyruvate happens at the end of cellular respiration. The pyruvate gets transformed into acetyl-CoA. Acetyl-CoA combines with the oxaloacetate to become citric acid. These three things are all intermediate. They're compounds that are made as you're going along the pathway from glucose to ATP. Hexokinase is an enzyme that is catalyzing, it's catalyzing the first step in glycolysis, but it's the enzyme, it's the thing that moves you from one intermediate to the next intermediate. So it's a critical part of the metabolic pathway, but it's not an intermediate. It's the enzyme that moves you from one intermediate to the next intermediate. So does that make sense to everybody? Yeah. And then finally, a cell whose membrane permeability is mainly to sodium will have a membrane voltage that is, and here we got, again, 78% people got it, but here we also have 20% of, actually 25% of you all got this wrong. And this is going to be critical for understanding everything we're going to do today. So if you are one of these negative or neutral characters here, make sure that you kind of get this idea. Sodium, very concentrated outside the cell. So if you open up a lot of sodium channels, the sodium rushes in. So cations coming onto the inside of the cell, what's that going to do to the membrane voltage?
It's going to be positive.
It's going to become more positive. If you get cations, positive charge coming into the cell, the voltage is becoming more positive. So that's going to be critical part of understanding stuff today is open up a sodium channel, the voltage goes up, we depolarize. So are there any questions about any of this stuff we've just covered here? This is all important, particularly the stuff about the ions in the membrane that are going to be coming up as we talk about neurons today. Okay, good. Alrighty, so we're going to do now is, I should also mention before we get into the actual material, let me go back to the schedule. I altered, you've probably noticed this is like a work in progress. I put here, notice this schedule is subject to change, and I've been changing it quite a bit as I get the reality check on how we're doing on everything. So the latest change here is, today is reflex action. This is Thursday, 9/24. I moved the nerve stimulation lab to after the exam. That way you don't have to think about getting ready for the nerve stimulation lab this weekend while you're also studying for your exam. Because the nerve stimulation lab actually has quite a bit of stuff to think about. It's, so after the exam on Tuesday, I will introduce the nerve stimulation lab, talk about what you have to think about to come up with your objectives and hypothesis and stuff like that. But don't worry about the nerve stimulation lab. until after the exam. So just kind of putting that out there in case. And then I should also mention, I don't know what the heck happened, but like 2 people for their reflex action pre-labs wrote hypotheses, objectives and hypotheses that were for the nerve stimulation lab. So that was weird. Part of me is thinking, so I'm not quite sure how that happened.
So in my lab manual or my lab book that I got from the school, it had different pages than the ones that you posted. So when I went and looked at the pages that you posted, it was totally different.
What do you mean the pages?
Like for the lab, for the reflex lab. Like the PDF pages that you post about the reflex labs were different than the ones that came in my book.
I almost accidentally did the nerve stimulation one too.
Because it's in order, it goes nerve stimulation and then reflex lab.
Oh, so the order's just switched.
Mm-hmm.
Interesting.
Okay.
Check it. Yeah, so definitely, because in that announcement I wrote, it talked about reflex lab. If you went to the module, it was all about the reflex lab. If you watched the video that I posted of me talking about things, it's all about whacking on the knee. So if everything in the module is all about hitting a knee with a little hammer, you know, it doesn't really make sense to write a pre-lab that has to do with electrical stimulation and thresholds and stuff like that. So that, yeah, that, I was kind of impressed. One of them was like, you know, it was impressive for the other lab, but it wasn't for the right lab. So anyway, let's now dig in to the neuron and graded potentials and action potentials and synapses and all of that good stuff. So here we go. I'm going to now share content. Okay, so this is kind of the beginning of the nervous system. We're going to introduce some of the really fundamental concepts of, you know, the neuron obviously is the most famous cell in the nervous system. There are other important cells that we're going to talk about, all this supporting cells, the glial cells and all that. But for today, we're just going to focus on the neuron, the kind of rock star excitable cells of the nervous system. And then we're going to talk about the different ways that they send messages and the ways that they communicate with each other. So that is today's goal. So let's just introduce the neuron. Wow. The neuron. The neuron is also known as just a nerve, like sometimes they're called nerve fibers because they're really long and thin. Typically, you know, and there are different forms of them. I'm going to draw the classic multipolar neuron, which is the most common kind. If you're in anatomy, you probably remember there's unipolar neurons and bipolar neurons. You know, for this class, we're not going to get into those kind of details. This is the most common, the kind of multipolar neuron. We'll talk about, again, just the amount of anatomy we need to talk about the physiology is the basic theme that we have here in this class. A basic neuron is got a cell body. The cell body is known as the soma. There's the little extensions where messages are typically coming in, which we call the dendrites. Dendrites just mean little branches. So again, the dendrites are these incoming messages. There's usually one main long extension where messages are going out, and that's the axon. The axons can be really long. Like you have like the motor neurons that operate like the muscles in your foot. The cell bodies are in your spinal cord, and yet they go all the way down to your foot. So you have, and the same thing for the sensory. If someone's tickling the bottom of your foot, those messages are coming up a neuron of a single cell. So you can have an axon that can be, you know, a meter long. So this can be super long. The axon originates at a part of the soma called the axon hillock, which is going to become important as we continue our discussion. The axon hillock, we're going to see, acts kind of like an integrator. It's kind of looking at all of the activity that's occurring at the neuron and deciding whether or not we need to send a message out the axon to the next chain of the neural pathway, or to the next link in the neural pathway. So axon hillock is going to be important. It's going to be where we kind of do the summation of all the activity across the entire neuron and decide whether or not to send a message out down the axon. This sigma just means sum. It's just adding in all the activity across this whole thing. We're going to talk about that in way more detail in a little bit. At the end of the axon, you have little branches. These are the telodendria. Telo meaning far away, like, you know, telescope is looking at things that are far away, like the stars. Telodendria are the little branches that are at the end of the axon, far away from the soma. And they have these little swellings at the end. These can be called synaptic synaptic knobs, or what I tend to like to use is boutons. Boutons, it's just like the French for little buttons. These are going to be what make the synapse with the next neuron, or with sometimes a muscle, a target. We'll talk about these in much more detail in a little bit when we talk about synapses, how one neuron talks to the next neuron. So we'll come back and meet the boutons in more detail. These, though, like if we have other neurons, they're going to be coming in and sending in little messages to another neuron. So this neuron is going to be getting messages from the little synaptic boutons from other neurons that are talking to it. So, you know, and this is going to be talking to some neuron over here, which is sending its message out someplace else. So we'll be looking at this. This is going to be these little synapses that are going to be these communications between one neuron and the next neuron. These are going to be our little synapses, which we're going to talk about in way more detail. Other things, before we leave just the basics of the neuron, we should talk a bit about the just practical nature of, you know, where do you actually have the genetic code to make proteins, make the stuff that the cell wants to make?
Nucleus.
In the nucleus.
In the nucleus?
Exactly, in the nucleus. And this is where we have the code. This is where we're going to have lots of rough endoplasmic reticulum making all sorts of stuff that the cell needs. There's actually, when people first were studying these, people didn't even know what, you know, endoplasmic reticulum was. They just knew that thing stained darkly inside the cell. They call it sometimes the Nissl substance. It just is a rough endoplasmic reticulum in here making lots of proteins. But then we need to get it out here. We need to get stuff that we make inside the cell down to the end here. And we just said that the axon might be 3 feet long. So so we need some way to move stuff a really long distance. So there's a couple of different ways that you can move things along this. We can have slow versus fast axonal transport. So slow axonal transport is just kind of flow of the cytoplasm, right? So that's kind of what you might think. It's just kind of the fluid is flowing, things are moving, diffusing. It's really slow. It's going to take a long time to go from one end here 3 feet down to get to the other end. So fast axonal transport is a system, it's a lot like a railway. So for fast axonal transport, you literally have microtubules running along the length. This is fast axonal transport. And you have these little proteins, they're called kinesins, that basically walk along and drag things. They use ATP. I kind of think about some old like Western movie where someone's on the railroad tracks with the pump car going chunk, 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 and they're kind of moving along the track. It's a lot like that. In fact, you can actually, I've seen little videos where they will attach little like beads onto the kinesin, and then they'll puff little bits of ATP, and you just see these things start walking along the microtubule. And so this moves along where you can actually get quite a bit, I forget how many millimeters per day, but you can actually get pretty far in, so this goes pretty fast. If you're moving towards the far end, towards the telodendria, then it's called anterograde, meaning forward. You can go backwards as well, which is called retrograde. So, sometimes you have stuff here that you want to bring back into the main cell body to recycle or to deal with. Part of the reason this is important to know is this system can get hijacked. Particularly, it's like, herpes. Herpes, if you catch herpes, whether either oral or genital herpes, that little herpes simplex virus, actually, you get it on your skin, which is where the neuron is actually kind of projecting to. It's either on your lips or on your genitalia, but those little viruses can actually hitch a ride and get into this fast axonal transport. Then they take up residence, and then they kind of live inside the cell bodies. And the cell bodies, for those sensory neurons, those are near your spine. So you've got the herpes virus kind of taking its little between outbreaks of herpes. The virus is just sitting inside the cell bodies of these neurons sitting near your spine, along your spinal cord. And when you have an outbreak, it's like those little viruses can hitch a ride on the anterograde train and end up back on the surface. And then you've got an outbreak, and now you've got little like cold sores or something showing up on your mouth or your genitals. So like, if you're wondering, like, how does herpes work? It actually works by hitching a ride on this fast axonal transport to get pulled into the cell body where it kind of lives when you're between outbreaks, and hitch a ride back out when you have an active outbreak. So this is, in general, fast exonal transport is good, helps the cell do everything it needs to do, but it does get hijacked, in this one particular example by kind of one of these nasties. So that is the neuron. The neuron kind of more as a cell. What we're going to talk about now is the neuron as a thing that's signaling things with electrical signals as well as with chemical signals. So let's do that. La, la, la. Here it is. I kind of made I'm going to keep myself on track. Introduce neuron. Check. Now we're going to talk about graded potentials versus action potentials.
One more question, Professor.
Uh-huh.
So where does that myelinated sheath and the nodes of Ranvier come into play in this?
So that is a good question. So we're going to look at myelin in much more detail soon, but in not all neurons, but some neurons that are optimized for very fast transmission, there are going to be these things in myelin sheaths, these insulating wrappings along the axon with little gaps. So the wrapping is going to be the myelin sheath. The little gaps are going to be the nodes of Ranvier. However, this is going to make more sense once we actually are talking about action potentials propagating down the axon. So I'll come back to this. When we look at action potentials, we'll talk about why it makes sense in some cases to have myelin and what the effect is on the rate that the action potentials propagate down the axon. It's basically a way to really speed things up. If you want to send messages really fast, you need to have this insulating layer of myelin, and we'll talk about why that's important in a little bit. Okie dokie. So graded potentials. Whoops. Versus action potentials. We call these APs. So potential meaning just voltage. Remember, membrane potential is just a synonym for membrane voltage. Whenever you see graded potentials, action potentials, it's meaning voltages, membrane voltages. There are two main kind of forms that these take, and they're related, they affect each other, but we're gonna look at them right now. We're gonna introduce them, and then we're gonna go into much more detail about both of them. First thing, graded potentials. These are continuously variable. These can be positive or negative. Larger or smaller. A graded potential can be the voltage goes up a bit, or it goes down a bit, or it goes up more, or it goes down more. So there's a whole, when we say continuously variable, that means you have a knob and you can turn it up more and more and more and less and less and less, or down even more and more negative or more and more positive. This is going to happen ultimately by having ions cross the membrane. Having more ions cross the membrane is going to create more of an electrical change. If they are anions moving in, it's going to be a negative change. If they're cations moving in, it's going to be a positive change. So graded potentials are ultimately going to be the result of ions crossing the membrane. And the more or less ions, it'll be more or less of an effect. If it's cations or anions moving in or out, it's going to be positive or negative kind of voltage change. So that's the idea of graded potentials. Action potentials are going to be a totally different thing. It's going to be this big chain reaction thing. These are always positive. And all or none. By all or none, we mean it's either running or it's not running. The best analogy to do here is like the fuse on a stick of dynamite, right? If you light the fuse, either the fuse is going or the fuse is not going. You can't have, oh, let's light the fuse and light it strongly, or let's light the fuse gently and have the fuse burn gently towards the stick of dynamite. It's either burning or it's not burning, right? Most people have a sense of lighting a fuse and zzz, going along. Or the other good metaphor would be simile, like or as. It's like a row of dominoes, and you push the first domino and the dominoes start knocking each other down. They're either knocking each other down and going along the row, or they're not going. You can't push it and have it go really fast, or push it the first one gently and they're each very slowly knocking each other down. It's either going or it's not going. That's why we say like all or none. The action potential is this chain reaction that's either going and running or it's not going. So those are a couple of the big differences. Always positive in all or none versus graded potentials, which they can be larger or smaller, they can be positive or negative. Other differences are where we find these. Graded potentials tend to be on the soma and the dendrites. So these are localized on the soma and dendrites of the neuron. Whereas the action potentials run along the axon. We are also going to see action potentials are critical parts of muscle contraction. So when we get to the muscular system in a month from now, we're going to see action potentials again running across the cell membranes on your muscle cells and stuff. But for right now, just think about them on the neurons. Then the final thing we'll add in here is graded potentials tend to be kind of local and short-lived. Local meaning they're just occurring in the one particular region where you've opened up some channels that let ions cross the membrane. So, if I have some neuron here and I've opened some channels here, there'll be some voltage change here, but then it'll stop if I stop the channels. Or if I open up some channels here, I'll have the voltage going up or down on the membrane here, but it's just right there. Action potentials, on the other hand, are this chain reaction that travel long distances. If you start an action potential here, it just moves and doesn't stop till it gets to the very end. It's this chain reaction that just moves on and on. So we have this idea, action potentials, travel long distances. And they're kind of long-lived. Once they start, they keep going. Again, it's like the idea of lighting a fuse. You light the fuse, but once you've lit it, it just keeps going. You don't have to keep doing anything. It just keeps burning until it gets to the end. Or you push that first domino, and now the dominoes keep falling and knocking each other down. You don't have to do anything. You just stand back and watch them go, right? Versus a graded potential, you do something, and if you stop doing it, it ends. And it only happened where you did it, right? If you like, I don't know, you push down on a mattress, it'll go down where your finger is, and you let go, and then the mattress will snap back and you're done. Whereas if you push the first domino, it keeps going and going and going. So that's like an action potential. It's like it goes on and on beyond whatever started it. And it keeps going till it hits the end. We're going to be looking at both of these, and we'll be looking at a lot of the details about how action potentials happen. Action potentials are going to rely on all sorts of these specialized voltage gated channels that we're going to be looking at in more detail. So we'll be spending more time looking at what exactly an action potential is, how it works, how it's propagated, what affects it. But let's start before we get there, let's start by spending a little more time looking at graded potentials. Because at some level, they're a little more straightforward and intuitive. So typically, graded potentials, if this is like some cell membrane, we have some channel here. Graded potentials typically happen, and here's some ion channel. Graded potentials usually happen because on some little part of the membrane, you're opening up an ion channel. So if I have, like, let's say I have potassium, actually not potassium, let's say I have sodiums coming in. Then my voltage is gonna, VM is gonna go up. I'm gonna have what we call depolarization. Let's say I have chloride coming in. My voltage is gonna go down. I'd have repolarization or maybe hyperpolarization. Or maybe if I open up channels and I have potassium running out, this is also gonna make the voltage go down, because if I have positives leaving, that's the voltage is going down, right? So graded potentials happen when you open some ion channel, ions are running in or out, and that's going to change the membrane voltage, the membrane potential. Typically, what's causing this in neurons is activity at a synapse, where one neuron is talking to another neuron. So we're going to talk about the synapse of a neuron right now. So how one neuron, communicates. Actually, I am going to erase that and type it so it's prettier. I'm going to do so we have one neuron, and it's going to be sending a message that is going to talk to the next neuron. So I'll have like, whoop. So let's say I have this neuron here. My little boutons with its dendrites. Then I'll have another neuron. I'll have my blue neuron. with its dendrites. And it's got an axon going off someplace else. So maybe I'll have a yellow neuron here. With its dendrites. And then I'll have other neurons. I'll have the green neuron, which is coming in over here, and its axon is now talking to this one. Or I can have another neuron, and I can actually have the bouton on the actual soma. So each of these little things I'm drawing here, these are my synapses. These are the little places where one neuron is talking to the next neuron. And there are cases where there's a direct electrical connection, where one neuron actually is electrically connected to another, what we call an electrical synapse. What kind of cell membrane proteins would we find if a neuron was actually directly electrically connected to another neuron?
Direct gated channel.
No.
Something. No.
Junctions.
Say it again.
Gap junctions.
Yeah, so Morgan got it, right? The gap junctions. It is possible to have gap junctions where one neuron is actually electrically connected to another, but it's not the most common way. So usually it's through a chemical synapse. So the neurons do not actually touch. So the neurons are close to each other, but they are not actually touching. So what we're going to do is look at this synapse and look at how it works, talk about the details a little bit. So this is going to be my presynaptic axon. So this is the axon, that process that's coming from one neuron heading to this place where it's going to talk to the next neuron. And then on this side, I'm going to have my postsynaptic dendrite. So are people kind of, the basic idea here is we're looking like so presynaptic axon, the little axon that is heading into the synapse. And then my postsynaptic dendrite, this is the neuron that is getting the message. So I am magnifying this greatly in this next page that we're looking at here. So we're seeing this neuron that is heading into the synapse, and then the neuron that is being talked to, that's on the other side. It's important to realize that these do not touch. There is what we call the synaptic cleft. It's dinky, but it's real. So it's about 50 nanometers or so. You know, so things can diffuse across pretty fast, but it means they're still not touching. What is a nanometer? Who can tell me what a nanometer is?
Isn't it 1
Micrometer
billion?
divided by 1,000.
Say it again.
1 billion.
Yeah, it's the 1/1 billionth. It's 10 to the minus ninth. So this is 50 times 10 to the minus ninth meters. It's really small. It's really small. So it's, you know, a nanometer is 1,000,000 times smaller than a millimeter. So it's an actual gap, but it's a very dinky gap, which is why things can diffuse across very, very efficiently. Other things we have to set up in our picture here. We are going to have synaptic vesicles here. And they are filled with different kinds of signaling molecules, the neurotransmitters. Neurotransmitters, they're just signaling molecules that are specifically found in the nervous system. Again, serotonin, norepinephrine, dopamine. We're going to talk about a variety of them in a little bit. I'm going to talk about some of the main ones you need to know, like acetylcholine and things like that. So there's a variety of neurotransmitters. There's over like 50 at this point, I think, that we know of. But for right now, just think about them as the signaling molecules that allow one neuron to talk to the next neuron. Obviously, if the next neuron is going to get the message from a signaling molecule, it needs receptors. So let's put receptors on our postsynaptic membrane here. Rack. So these are my receptors that will bind the neurotransmitter. Again, there's obviously, we've talked about, there's all sorts of different neurotransmitter type receptors. We looked particularly at serotonin receptors a little bit before, but there's obviously receptors that will bind dopamine or bind adrenaline or bind whatever. So these are the appropriate receptors for whatever neurotransmitter is sitting in these little vesicles. And now we need to add one more piece into this. We're going to put in some voltage-gated calcium channels in our presynaptic terminal here. So this is my, remember this thing here, this is like my bouton, right? My little terminal. I'm going to put in some calcium channels. These are going to be voltage-gated calcium channels. So now we're set. Now we are set here. So the basic, so I'm gonna like do a little hand waving with the action potential since we haven't actually talked about the details of the action potential yet. So we're going to assume that there is an action potential, one of these big excitatory all or nothing signals that is coming down the neuron. Some action potential is running down. So action potential runs down the axon, gets to the very end, gets to the little synapse that we're talking about here. Once we get to that end, the voltage is going to go up. The action potential is always positive. So we go and we look at this picture we just said here, and we've got our little voltage gated calcium channels. So what is going to happen when the action potential reaches this little bouton, this little end of the axon here? What is a voltage gated channel? What's the definition of a voltage gated channel? I know people know this. Come on.
It's going to open or close based on the like the voltage of the membrane.
Exactly, exactly. So basically the first thing that happens is the action potential, reaches the bouton or the axon terminal or whatever you want to call it. That is going to cause the calcium channels to open. So the calcium, they're calcium 2+. And calcium enters the cell. So that is the first step in kind of triggering this whole thing, is that action potential reaches the bouton, the calcium channels open and the calcium enters in. And we are not going to get into the detail. People have actually figured out a lot of the mechanics here. We are just going to do a little hand waving because we need to draw, we need to kind of figure out where to focus, where not. I'm going to say basically this triggers the exocytosis of the neurotransmitter. So we had this voltage getting all positive coming down here. That triggered the opening of these calcium channels, and now calcium is coming in. Once calcium comes in, that is going to trigger the exocytosis, where these vesicles are going to fuse with the membrane here and dump the neurotransmitter into the cleft. So now the neurotransmitter is in the cleft. What's going to happen next?
It diffuses across the synaptic core.
Exactly.
And we'll bind the receptors.
Exactly. So the next thing that happens here in like kind of part 4, neurotransmitter diffuses across cleft and binds to receptors on postsynaptic membrane. And what's going to happen next?
I have a quick question. Is it the vesicles that are leaving or the neurotransmitters like in the vesicles that are being
Yeah, no,
transport.
the exocytosis, the vesicle fuses, so the membrane just becomes part of the membrane of the cell and
Okay.
dumps out the contents. So it's just the contents, just the neurotransmitter that's getting dumped.
Thank you.
What happens next?
Another channels, gated channels are going to open.
Yeah, exactly. Exactly. This is going to cause ion channels to open in the postsynaptic membrane. Right? It might be through, let me make sure this is high enough so you can actually, can you, is it off the bottom of the screen or can you read it?
I can't see it.
It looks good.
Okay, so, this might be through directly gated channels like we saw with the nicotinic cholinergic receptor or those GABA receptors, or it might be through a second messenger pathway like we've been talking about. But one way or another, we're open up ion channels. And when we open up the ion channels and ions are gonna cross this membrane, it's gonna cause a graded potential in the postsynaptic membrane. Right? If we have ions crossing the membrane, that's causing a voltage change. The voltage is going up or down. Is the voltage gonna go up or down? What do you think? When we trigger a synapse, will the voltage go up or down in the postsynaptic neuron?
Will go up, right?
It depends
Depends on the ion.
It depends on the ion. It depends, it might go up or it might go down. There are inhibitory synapses. There are synapses that make the next neuron less excitable, like by opening chloride channels. If that neurotransmitter was GABA, it's going to open chloride channels, which make the next neuron more negative, less excitable. So we talk about inhibitory versus excitatory postsynaptic potentials. So a EPSP, excitatory postsynaptic potential, you depolarize the membrane. Right, you become more positive. That would be like opening sodium channels, letting in sodium. But it's possible to have what we call an IPSP, inhibitory postsynaptic potential. So VM goes down. Example, like letting chloride enter. Right, so we can either make the thing more positive or more negative at a synapse. So it's important to remember that synaptic activity doesn't necessarily excite the target. It actually might inhibit the target. I mean, that's actually an important thing to remember. Like, you actually can get real issues. If you stop inhibition, you can actually create seizures and stuff because things get overexcited. So having proper inhibition is important as well as having proper exciting when it's appropriate. So EPSP versus IPSP, you gotta remember those two things. Are there any questions about what we mean about that? The last thing we'll talk about before we move on to the action potentials is the idea of summation. So summation means adding up. There's two different ways summation can happen, and we'll look at both of them right now. So let's assume here's some neuron. And then let's say I have two other neurons talking to it. Let's start with one neuron. If I have one neuron talking to it, and let's say this neuron sends a message down its axon, releases some neurotransmitter here, some ions, let's say it's excitatory. It lets in some sodium and then the voltage goes up, right? So here, let's say this is V sub M. If I'm looking right here at this membrane here. You know, I have some action potential comes here. The voltage on my membrane is going to go up a little bit because I opened up some sodium channels and the voltage went up. And let's say I send another action potential down here. It'll happen again. Open the channels again, voltage goes up and then comes back down. And again, graded potentials are short-lived. They start, but then they end after the excitation is gone. What happens though, if we start sending action potential after action potential in quick succession down this neuron and keep releasing neurotransmitter, and we keep the ion gates open, what happens is the voltage goes up, but before it has a chance to drop back down, even more sodium comes in and it gets even more positive, and they're still open because we're still sending more messages down and leaving these receptors bound and keeping the ion channels open. So we can actually get a much larger voltage increase than we would have from a single thing. This is called temporal summation, if this is happening. Temporal meaning within time. So it's just a single synapse, but if you keep firing off that synapse in quick succession, the overall effect will start adding and you'll have an even greater voltage change at the target membrane. This is going to be important next week when we start coming up with the hypotheses for our nerve stimulation lab. This idea that if you are constantly exciting this, you start adding up the effect at the target.
One question, Professor. The channels will remain open, or they will open, close, and the ion will accumulate?
Yeah, exactly. The ion accumulates rather than kind of resolving and going back to the resting.
Okay. Thank you.
Yeah. All right, so that is temporal summation. Another kind of summation that we have is called spatial summation. So let's say I have one neuron talking here. Let's give the other one, we'll make it blue. It has a synapse that's nearby. And in this case, let me give myself some room. So I'm going to be looking at the membrane potential right here. It's supposed to be my eye. I'm looking at the membrane potential right around where these synapses are happening. If I fire off the green, there'll be a certain change in membrane potential. It'll go up and come back down. If I fire off just the blue, it will also have some depolarization and come back down. Spatial summation is if I fire off these two things that are close in space at the same time. So I am now going to fire off both the blue and the green together. And the overall effect is going to be additive. So this is what we call spatial summation. Both of these are firing off together close in space. So this one's opening up its channels here, this one's opening up its channels, and they're right nearby so they can add together to have an overall larger accumulation of ions in the membrane at that area. So spatial summation, these fire off together. Fire together in same space. Near same space, I should say. Near same space of membrane. This is going to be important a lot when we get further on into the realities of how the nervous system works. The idea of synchrony, having different neurons firing together, and then the nervous system being able to actually notice that and respond to that. That can happen through this idea of spatial summation. If lots of neurons, if these neurons are just firing randomly, they're never going to add up. You're never going to have more than just this smaller change in the membrane voltage here because they're not firing at the same time. But if they are synchronous, if they're firing together in unison, in lockstep, you'll have a much larger voltage change, a much larger effect at the target, which then is going to be important for triggering, signals that continue down to the next phases of the neural networks. So having spatial summation is important to be able to detect like synchrony, to detect when things are kind of converging and firing together. Does that make sense? All right. So,
can you go over one more time the both type of summations?
Yeah.
Please.
So, for sure. So there's two ways these graded potentials tend to add together, summate. The first one I mentioned is just relevant to looking at a single synapse. If you have a single synapse and you fire it, there'll be some particular, change in membrane voltage, some graded potential that's resulting of channels opening, ions crossing the membrane, changing the voltage. If you fire that same synapse in quick succession, that's why it's called temporal summation, kind of, in time, then as the channels keep opening again and again quickly before the last change has had a chance to resolve, we start accumulating more and more ions across the membrane, having a larger and larger voltage change that shows up. So temporal summation, one synapse fired in quick succession results in a much larger overall effect, much larger graded potential happening at that target membrane. So that's temporal summation. Spatial summation is thinking about multiple neurons that are all kind of having synapses nearby each other. And if they fire together, you can add their overall effect because they are causing ions to come in the same area of the membrane. And since they're close by, the effects of the membrane voltage are going to add if they're running together. So I don't know if does that help, Jamba?
Yes, thank you.
Okay. All right. Any other questions about this? All right, so we need to finish up with action potentials, which is going to take a little bit of time. What I think we should do, let's take a break. We're going to tie up the action potentials. That will tie up all the lecture material. And then we'll take another little break, and then we will get into the reflex lab. But let's take a break so people have a chance to let this sink in. And then we'll talk about what the heck actually do we mean here by this action potential coming in. What is an action potential? So let's take a break and return. It's 9:33. So let's return at 10 a.m. And at 10 a.m. we will talk about action potentials. But here we go. Action potentials. Again, the idea here is there are these kind of all or nothing chain reactions that travel long distances, again, can move from the soma in your spinal cord all the way down to the tip of your toe in a fraction of a second. You get these big, long distance, fast signals moving around. We are also going to, again, see these later on when we get to the muscular system, spreading across the cell membranes of the muscle fibers to activate muscle contraction and things like that. To understand action potentials, you need to understand two specialized membrane proteins, two voltage-gated ion channels that are found specifically in excitable membrane, in the axons of neurons, in the sarcolemmas of muscle cells. So I am going to introduce the two special voltage-gated channels that you need to be familiar with, and that will be necessary to understand how action potentials work. So, two voltage-gated channels. These are in the axon membrane. Right, you don't find these in the dendrites or in the soma. These are about creating this action potential that goes along the axon. Again, we'll find them again when we get to the muscles, but for the neurons, they're in the axon. So there's going to be the voltage voltage gated potassium channel and the voltage gated sodium channel. They are both similar in that they are closed when the cell is at its resting. So these are closed at V rest. Which is around, again, minus 70 millivolts. So when the cell is at rest, these are closed. They will open when the cell membrane rises above the threshold of minus 55 millivolts. So far, they have the same story. They're in the axon membrane. They're closed when the cell's at rest. They open when the cell has a membrane voltage, or when the membrane voltage goes minus 55 millivolts or above. But now we're going to come into some big differences between the two. The voltage gate, so let's draw a line here. The voltage gated sodium channel, no, potassium channels, these are slow. They open slow, takes about a millisecond. Millisecond, again, it sounds fast. What is a millisecond? How much is a millisecond?
1/1000th of a second.
1/1000th of a second. But that's actually pretty slow in the big picture of things. Especially when again, when you're talking about computers and stuff, you're talking about trillionths of seconds and stuff. And we're going to talk more about that, like how the heck the nervous system is able to do what it does when the processes in the cells are so pokey compared to what you'd expect for really fast processing. They also close slow. Also, so I always think of them as like, those kill the rabbit, which way did he go towards? It's like, ooh, the membrane just came above minus 55. It's like, whoa, the membrane voltage is above 55. It's time to open up. And then the voltage drops back down. It's like, oh, wow, look, the voltage has dropped back down. It's time to close. So that lag is going to be important when we see how the action potential forms, because there's going to be other stuff happening much faster that's going to leave these little channels in the dust, particularly the sodium channel. The sodium channel opens fast. So these pretty much snap open pretty much instantly. For our purposes, as soon as the voltage hits, membrane voltage hits minus 55 and above, they're open. Imagine they're open. Again, these guys aren't going to open for a whole millisecond after this. So there's going to be this millisecond where the sodium conductance is really high and the potassium conductance hasn't changed at all. The sodium channels are a little more complicated in that they have this inactivation gate. So these then will inactivate after 1 millisecond. So this is different than closing. This is basically when they open, they spring open, but then there's this automatic thing that will shut the channel even though we're still above threshold. So this is still above threshold. So it's basically they have a limited time they stay open. You know, you activate them, they spring open almost instantly, but then after a millisecond, they close on you, even though the voltage of the membrane is still high. So these are our two main players. These slow potassium channels that will start opening at -55, but take a millisecond to really open fully. These fast sodium channels, which will open almost instantly when the voltage of the membrane hits -55, but after about a millisecond, will close down on you until they reset. You know, and they're not going to reset until the membrane potential comes down below -55 and everything has a chance to kind of go back to its resting. So are there any questions about these two channels? Because these are going to create the action potential. Okay. So now let's look at one particular piece of membrane. So we're going to look, let's just look at one particular piece of membrane that has these channels. V sub M. Now I'm going to give it numbers. So here is my membrane potential. And if you remember, a cell that's at rest is going to be around -70. I'm also going to put that threshold here just so you can see it more clearly. -55. So that's, we have to pay attention to that because that's when those channels are going to wake up. And so a cell, I said, is -70, but it can change depending on different, you know, the graded potentials. If you're looking at a cell with all these synapses coming on it, some neuron, the neuron is, in fact, let's just draw that. Here is my neuron. Here is my axon. Here's my axon hillock. And here are all of these synapses coming in. You know, and these are firing off, and they might be excitatory postsynaptic potentials or inhibitory postsynaptic potentials. So these are all working together to make the voltage go up a little or go down a little. So if I'm looking right here at this axon hillock, the beginning of the axon, you know, I'm at -70, but it's actually kind of going up and down around -70 because if there's a bunch of positive, you know, excitatory postsynaptic potentials, the voltage is going to get higher. There's more negative postsynaptic potentials, the inhibitory postsynaptic potentials. It's going to drive the overall, you know, that's what we're looking at here. We're looking at the sum of all the EPSPs and IPSPs. The sum, the addition of all the excitatory postsynaptic, inhibitory postsynaptic potentials here at the hillock. You know, excitatory ones are making the overall sum higher. Inhibitory ones are pulling the overall sum lower. Does that make sense? So if we go back to this picture here, this is actually kind of wiggling up and down. The membrane voltage is going up and down based upon the activity of all those synapses that are talking to the neuron. So here, let's look specifically, we're at the axon hillock, which is the beginning of the whole thing. So at some point, let's say there's enough excitatory postsynaptic potentials that it crosses the boundary. We hit threshold. At this point, what is going to happen as soon as we are above minus 55 millivolts?
The channels will open.
And which ones in particular are going to be the ones that snap open fast?
The sodium channels.
Exactly. So sodium channels, all of a sudden, we've got a huge sodium conductance. All these sodium channels have just flipped open. What's going to happen to the voltage on the membrane as all the sodium rushes in and we start approaching that equilibrium potential for sodium?
It'll increase.
Say it again.
It'll increase.
It's going to increase, and it's going to go up, and it's going to go up, you know, close to, you know, maybe up to plus 40 or something. And this, I should put some time on this. Let's say this is 1, 2, 3, 4, 5 milliseconds. Basically, it's going to rush up. You know, this is going to be, you know, fast opening sodium channels. Right, the sodium channels are open, and as the sodium rushes in, the voltage is going to rush up towards like plus 40, 50, something like that. But then, oh my God, we're about a millisecond later, what's going to happen?
The potassium channel opens.
So potassium channel opens, and what else happens?
It closes.
It gets inactivated, the sodium channels.
Sodium channel.
So sodium channels inactivate. Potassium channels open. So now we have a really high potassium conductance and no sodium conductance. What is the voltage going to do now if we have mainly potassium conductance?
It continues to increase.
Goes down.
So remember, potassium, remember if you remember from last class, if a cell is just permeable to potassium, that's where you go down to minus 90 millivolts because you've got such a higher concentration outside than inside. The potassium wants to leave. And also if it's really positive inside, that's also going to make the potassium want to leave. But basically the potassium leaves. And not only does it go down to back where it was, because we now have a higher than normal potassium conductance, it goes even lower. We have this hyperpolarization going on here. And then after about another millisecond, after the potassium channels finally realize, oh wait, we're down negative, we should close, then we kind of go back to the normal resting. So there's this kind of undershoot where we have the potassium channels are still open and we have more than normal potassium conductance, which means we're even closer to minus 90 than we would be at resting. So this is a classic looking action potential. Fast opening sodium channels when we hit the threshold means the voltage is going to rocket up to about plus 40 or something. Then after a millisecond, sodium channels close, potassium channels open. Now we come back down. We actually undershoot because now we have the potassium channels actually stay open for about a millisecond even after we're lower than the threshold. But then after about a millisecond, everything's kind of rested and back to normal. So does this make sense? This is kind of looking at an action potential on one little piece of membrane, specifically at the hillock here. But we're going to be watching what happens as this chain reaction occurs. But you got to kind of understand this piece first.
One question, Professor. When potassium goes into the cell, potassium's channels open, it's going to get hyperpolarized.
Exactly.
Beyond, not depolarized.
It will
repolarize.
It'll repolarize and then continue even more negative because even more potassium is coming in than normal.
Okay. Okay, perfect. Thank you.
And that is going to actually, okay, and then there's two things that are going on here. So, so I got my stylus, come on. I'll just talk about it while my stylus recharges briefly. So we talked about how they open at minus 55. When you get down below minus 55, they have to kind of reset. It takes about a millisecond before the channels are really ready to go again. So no matter what kind of excitation you do, the channels cannot regenerate another action potential for at least about a millisecond after one action potential has happened. So for about 1 millisecond, we are going to have what's called the absolute refractory period. Let's spell absolute properly here. Absolute refractory period. This is the time when it is impossible to fire off another action potential. We're going to see that this is actually going to be very important in a few moments. So right after you fire off one action potential, you can't fire one immediately. You have to wait at least a millisecond before the membrane kind of recuperates and is ready to go again. After that, because of that hyperpolarization, the voltage tends to stay a little lower than normal resting for a little while. You can fire off an action potential here, but it's harder because the membrane is below its normal resting because we're hyperpolarized, which means you need more excitation to come up to the threshold. So there is this thing called relative refractory period. Maybe I should I'm going to go on to another page just to type these out. So absolute refractory period. So for 1 millisecond after the action potential, the channels are resetting and it is impossible to fire off another action potential. So that's going to be important. After you fire off one action potential, you have to wait about a millisecond before you can fire the next one off using that same piece of membrane with those channels. The relative refractory period. So after the absolute refractory period, the membrane is still hyperpolarized due to the extra potassium channels that are open. And therefore, it requires more excitation than normal to fire an action potential. So relative refractory period, it's possible to fire off an action potential, but it's harder. You have to excite the membrane more than normal because you have these lagging potassium channels that haven't all finished closing yet. So they're pulling the voltage down, making it harder to reach threshold and fire off the next action potential. So does that make sense? So again, if we kind of go, heck. Why am I? Oh, there it is. If we go back here, this relative refractory period is while the membrane is coming back to its resting. And it's still kind of hyperpolarized because those pokey potassium channels haven't all closed and gone back to their resting. So is this clear, crystal clear?
I have a question.
Yeah.
For me, it'd be helpful, I think, if you went over again, like why, when the potassium channel is open, it goes into the hyperpolarization. Like I understand, I just am trying to put the pieces together just because I'm getting a little confused because it is a positive.
All right. So if we go back to last class, we said, pretend only potassium conductance. And we had 5 millimolar, actually, what am I saying? Potassium inside is 150 millimolar. Potassium outside is 5 millimolar. So if it's only potassium conductance, the potassiums all want to leave because of their concentration gradient. As they leave, it becomes more positive here, more negative inside. And that keeps happening until we hit this V equilibrium for potassium is around minus 90 millivolts. So if you have a cell that is just purely potassium conductance, it's going to equilibrate at about minus 90 millivolts membrane potential. So we said in an actual neuron at rest, it's not purely potassium, it's mainly potassium. So rather than this, it's, V rest is around minus 70 millivolts. But when we have this excess potassium conductance from these voltage gated potassium channels that have opened in the action potential, we're getting closer to this. So normally V rest, it's potassium and some other stuff, but in that relative refractory period, we have excess potassium than normal, so we're closer to this minus 90 than normal. Does that make it clearer? Kind of?
Yeah, it does. Thank you. I think I just have to go through it a few times, just because I guess it was getting confusing for me where it makes sense that the potassium would leave in that first step because it's 150 inside, 5 outside, and then, but it's because of that next step because it has to balance out the positive negative.
Yeah.
Yeah.
The equilibrium happens when there's a balance between the voltage and the and the chemical force due to diffusion.
Right. Thank you for going through that.
Yeah. Yeah. Maybe depending on how much, yeah, maybe I'll show a little video after we've done all this that might make that clearer as well. All right, so let's get back to where we were. So other questions about what's going on here. Again, in a nutshell, as long as the voltage on the membrane is below threshold, everything stays quiet. As soon as we hit threshold, we start this whole big automatic chain reaction that's going to cause the voltage to spike up to like plus 40 millivolts or so, but then after a millisecond, plummet back down. That's the action potential. Okay, now we're going to look at it in the context of the whole neuron and the axon, which is completely covered with these channels that have this behavior. So here is my axon going along. Here's my neuron. And everything we've just talked about was happening right here at the hillock. The voltage was going up and down. We hit threshold and bingo, we had an action potential. But now what we have to think about is like, oh my God, it just got super positive right here. But the next little part of the membrane has the same channels. So what's going to happen to the voltage gated channels on the next little piece of the membrane as the action potential happens right there?
It will also open.
They're going to open. And all of a sudden, as this becomes more positive, the action potential will appear on the next little part of the membrane. You know, as this little part is resolving, this will go away, but now the action potential is a little further down the road. But now when it's positive there, this depolarization is going to open up the channels on the next little part of the membrane. And so all of a sudden, the action potential will appear there, just as we have the action potential ending there. And then as it's positive, whack, as it's positive there, the next little part of the membrane is becoming positive, and those channels are going to open, and that's going to cause an action potential on that little part of the membrane, just as the action potential was resolving there. So the basic idea, I'm not going to keep doing it, this thing is zipping along. It's going, remember I said it's like lighting a fuse, and it just keeps going. Each time one little part lights up, that is going to trigger the next little part to light up, and the next little part, and the next little part, and it's going to go all the way till it hits the synaptic bouton and causes the exocytosis of the neurotransmitter. So does that make sense? It's like
the action potential makes the next part of the axon more positive, and then it triggers an action potential.
Exactly. As the membrane here becomes more positive due to this action potential, it's causing the next little section to become above threshold. As soon as this part of the membrane becomes above threshold, it's the same story. That becomes an action potential there. And as this becomes an action potential, the next little part becomes positive here. So that has an action potential. You know, meanwhile, you know, on the backside, these things resolve because they last about a millisecond, then they disappear. So if you just kind of looked at it as the voltage through time, it looks like this wave of voltage excitation is traveling along the membrane. Again, traveling long distances, all or none. There's no, I mean, at least to a first approximation, there's no such thing as a strong action potential or a weak action potential. It's either going or it's not going. So are there questions about this? This kind of sorry, this chain reaction going on here. So we could ask the question, in fact, I am going to ask the question, how come when this action potential is going right here, it's exciting the membrane on either side of it, really. How come the action potential doesn't actually re-excite the membrane from whence it came and start just heading it back again? Because we're saying, oh, we're exciting here, which is going to cause this next little part to have an action potential. But how come we don't actually have the action potential just re-excite the membrane that just excited it and start sending it back towards the cell body?
Because of the inactivation of the sodium?
Because of the refractory period?
Yeah, exactly.
Ah, it's the refractory.
That absolute refractory period. I'll type it out because it makes it just easier to do it. Basically, absolute refractory period, prevents the, previous part from getting excited and keeps the action potential moving forward, which we call anterograde. And we could say prevents retrograde. So if we didn't have that absolute refractory period, whenever you had an action potential like this, you would have the problem of it's exciting the next little segment, but it could also excite the segment that just caused it to happen. So because of the absolute refractory period, by the time this membrane is ready to go again, this action potential is down the highway and is no longer keeping the voltage high. So this is resting and just waiting for the next thing to happen. So the absolute refractory period means we can't automatically instantly excite the thing that just had the action potential. And again, by the time the action potential has traveled further down, this is now ready to go again, but the excitation is far away and is not going to cause mischief, sending messages backwards. That makes sense? All right. So the last thing we need to talk about to kind of tie up our story, and before I say this, let me reiterate this idea of the action potential is getting ultimately triggered by having the hillock rise above threshold. And the hillock rises above threshold based on all the activity of the synapses that are talking to this neuron. So again, the hillock we usually talk about as being kind of a integrator. It's integrating, it's summing. This is just a Greek letter sigma. It's what you use to do mathematical summation. The sum of all the EPSPs and IPSPs. All of the activity, all the synapses that are either taking the membrane voltage up or taking the membrane voltage down. At some level, the sum of all that is either going to be enough to bring us above -55 or not. If this becomes greater than -55 millivolts, then we trigger an action potential, which is going to go down the axon and send a message to the next part of the neural network. So, you can again, this kind of integrator, maybe you can integrate. So it kind of sums together all of the activity that's happening. This gives you an idea of why taking Valium or Xanax or any of those benzodiazepines makes you kind of less excitable, why it's anxiolytic, why it makes you less anxious or maybe even puts you to sleep. Xanax, we talked about all the benzodiazepines increase activity at those GABA receptors that let in chloride. So when you are taking Xanax or Valium, you are having more EPSPs going on, not IPSPs, more inhibitory postsynaptic potentials. You have more activity across the neurons where you have chloride coming in and the membrane voltage is going down, which means that this sum is going to be harder to bring up to threshold if you've got excess activity of chloride channels bringing the voltage down, which means all your neurons are less excitable, which means you are less excitable, right? So the way the benzodiazepines work is just by increasing these IPSPs, increasing the inhibitory postsynaptic potentials, by having more chloride coming into the neurons, lowering their voltage, pulling the axon hillock further down away from threshold, making it harder to bring it up to threshold and trigger an action potential. Does that make sense? So, okay, the last thing we need to talk about is the speed, how fast action potentials send the message down the axon. So velocity of action potentials. There are a few different things that affect the speed of action potentials. So one of the problem with membranes in general is, you know, if you have a voltage difference across, you actually can have, you know, electrical current, and eventually the voltage difference dissipates. And one way to increase, if you want to increase the voltage, I mean the velocity of an action potential, you can increase the diameter of the axon. If you increase the diameter of the axon, the voltage stays higher and you don't have to use as many channels to keep the voltage up. We see this in our own axons. We see this to the extreme. Extreme example. The squid giant axon. So in a squid, you know, if you spook a squid, it goes, it's gonna like disappear and squirt you with some ink. It's fast. It needs to have really fast reflexes so it doesn't get eaten. The way that it has evolved to send the message super fast from its sensors to its muscles is by having this really big axon. It's literally, the axon is 1 millimeter in diameter. Really, really wide. I should mention, I remember when I first was learning about this, I was thinking it was a giant squid or something. This is just like your calamari squid, the little bitty squid, but it has a giant axon. The reason why the squid giant axon was so critical for us understanding this is how the guys who first discovered this actually were able to discover all the stuff we're talking about. They would literally take the squid giant axon and put it in a little dish. So these are guys, this was in the 50s. These two guys named Hodgkin and Huxley. Wait, Hodgkin and Huxley. These are the guys that originally discovered this whole action potential and how it worked with these voltage gated channels. They took a squid giant axon and they just tested it to death with all these different internal solutions, external solutions. And they could just like, okay, what if we put a certain molarity of potassium in here versus out here, and what are the electrical properties? Now let's take a little squeegee and squeeze it out, and let's put a syringe and let's fill it with some potassium solution and see how it acts now. And let's take a squeegee and squeeze it out and put it back in. And so they actually literally just had this thing and they played with it and tried all these different solutions inside and outside. They came up with their theory about the activity of the channels. You know, they hypothesized this that we're talking about here. It literally, this was before computers. This is in the 50s. So they had these differential equations that they used that would describe the activity of these, that would predict the voltage changes in the membrane. They literally spent an entire winter, like they came up with this model and they spent months with adding machines. I don't know if people even know what these things are anymore. These mechanical things. They did all this numerical integration using these adding machines that finally validated that their model, that these membrane channels with these properties would actually lead to the action potential that was observed. And that was how they actually figured it out. You know, now you just click with a mouse and you have the model in your computer and it goes show. You know, computers now are really freaking fast. But back then it took them months to actually validate their model. So this one way to get faster action potentials is to use larger axons. But again, the problem is typically, like if I'm looking at, let me look, let's say this is V sub M. You know, and let's say I have my action, here we're just looking at along the different parts of the membrane. Let's say I have the action potential happens here, the voltage goes up in this part of the membrane, but it's a local thing. It's going to kind of fade off pretty quick. So if I want the action potential to keep going, I need to have more channels right here and re-excite it again so it stays up. And I need to make sure that I have more channels right there so it keeps going up. I need to make sure I have channels really all along the way, and the channels have to keep rejuvenating, recreating this action potential. So it's like boom, 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 boom. So at each part of the membrane, we need to make sure we have the channels opening and creating a new action potential before the membrane potential drops down and the action potential disappears. This means that it takes a while to get to the end. If we're just relying on the action potential being held up by channel after channel after channel, this might go, you know, the slower ones are maybe 3 to 4 meters per second. Not that fast. You know, if these were the only kinds of axons you had, you'd be in trouble. You like put your finger on a hot plate and the message will take literally like whoosh to your brain, and your brain's processing, oh, I'm in danger. And then whoosh, back to your muscles. By the time there's a signal for your muscles, your hands turn to embers or something. So we need something way faster, particularly for these sensory neurons, for these motor neurons. So the message flies into your brain really fast. You can process it and it flies back really fast so you can activate your muscles and have some response. So the way we can increase the speed dramatically is by using myelin. And again, the basic idea here, like think about, you know, I'll use the analogy of like if we had like a battery, let's say we have a battery here, you know, some battery, and there's some wire, and there's some light bulb. Let's say there's a light bulb that's running off the battery. You know, batteries, and in batteries, you actually think about the negative charges, the electrons that are flowing. So we have electrons flowing. The electrons flow out one end. There's a, you know, a potential, a voltage. So the electrons flow, they go through the filament, heat it up, it glows, and they come in. So we have a flow of electricity. But what if we take this and we put it in salt water, which is your whole body's freaking salt water. If you put it in salt water, then the electrons leave and they're gonna, oh, salt water, that's conductive. I can just short circuit. There's no reason to go through the light here. So the light bulb doesn't light because the electricity has gone through the salt water and dissipated. So how can I actually have my little circuit work in salt water and make sure that the electrons don't go through the salt water and stay in the wire and go to the light bulb?
For insulation?
I use some kind of insulation, you know, put some plastic around the wire so the electrons stay in the wire and don't get out and dissipate. So it's the same basic idea with your axons. If you don't have any insulation, you have this voltage change, but then the voltage basically dissipates very quickly and drops back down. That's why you need to have channels at every little segment of the membrane to keep rejuvenating, keep pushing it back up. However, if we have myelin, myelin is, we're going to talk about myelin in more detail next week after we introduce Schwann cells and oligodendrocytes. But for right now, let's just say it's this specialized insulating membrane stuff that wraps around the axon. And we get these things called myelin sheaths. So the myelin sheath, the thing that a myelin sheath does is when the voltage goes up, it takes much longer to drop down. So this would be like with myelin. When we didn't have myelin, it went up, but it kind of dissipates really quick. So without myelin, the voltage change dissipates, and you need to make sure you have more channels right away to keep it up. When you do have myelin, the voltage stays high much longer. You eventually do need to rejuvenate it, but we have these little nodes of Ranvier, you know, with our sodium and potassium channels, you know, to rejuvenate the action potential. But these only have to happen like, you know, maybe every 2 millimeters. This is about 2 millimeters. So this means instead of going like, in fact, I can show you like, here's my axon, here's my target. If I don't have myelin, I got to go rejuvenate every little section. It's like, open the sodium, open the sodium, open the sodium. Don't let the oxygen potential die. Open the sodium, open the sodium, open the sodium, open the sodium, open the sodium. Finally gets to the other end. If I have myelin, the signal stays high. We only have to rejuvenate it at these little nodes. So it goes like boom, 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 done. We have like the action potential velocity is more like 100 meters per second now. So like your fast pain fibers, the motor, skeletal motor neurons that send messages to your skeletal muscle, those are all going to be large diameter myelinated fibers. To send the messages really fast. You know, there are other things where you don't care if the message goes so fast, so why waste resources? So you have a whole range of neurons. Some of them are fast, some of them are slow, some of them are myelinated, some are unmyelinated. But it's important to understand that if you want a really fast message, you need to use a myelinated fiber. Sometimes, if you think about what is the function of myelin, what is, in a nutshell, what is the function of myelin?
It insulates.
Insulates, and why? To what end?
Makes the action potential faster.
Yeah, exactly. To increase the velocity of the action potential there. And we'll see a little more detail about the cells that make the myelin and some other roles the myelin can play. But for right now, this is what you should be thinking about. You should think about myelin, these insulation around the axon that allows you to only have to rejuvenate the signal a few times at the nodes, therefore creating a much, much faster signal. Sometimes they call this saltatory conduction. Saltare is like to jump. It's because it's like the signal is jumping from node to node. It's like the action potential started at the hillock. Oh no, it's there. No, it's there. It's there. Oh, it's home. It's gotten to its destination. Kind of like jumping. It's like chuck, 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 chuck. Instead of with no myelin, in which case it's like it's here and now 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 it's here. Kind of slowly moving along, and it's a little slow. So questions, comments about any of this?
Earlier you said that the larger the diameter, the faster the action potential. Can you just explain why that is?
It's the same kind of idea. This drop-off does not happen as precipitously if you have a large diameter. So you don't need to keep rejuvenating the signal as often. So it's another way to keep the signal from dissipating, but you have to have a much fatter axon. This lets you have a thin axon, but still keep the signal from dissipating.
Professor, one question. In the axons that are not, that has no myelin, the action potential goes from, it's slow because it goes from channel to channel. So channels open, close, open, close, open, close.
Correct.
Through the whole axon. That's why it's slow.
Right.
Okay. Okay.
It just,
thank you.
You have to, instead of just rejuvenating it at the nodes of Ranvier, every next little segment of the axon needs to have its own axons opening to keep the signal alive. So example, one of the kind of horrible autoimmune diseases that you can get is multiple sclerosis. Multiple sclerosis is an immune disease where your immune system attacks your myelin. So all of a sudden, the myelin sheaths are disappearing because your immune system's getting rid of them. What is that going to do to these signals going down the axons?
Slows them and lowers mobility. A lot of other things.
Right, so it's a couple of things. There's one thing is it can actually just prevent the signal from going, right? If the signal drops too low before the next channels are there, the signal might die out and never even get to the other end. You know, and typically the body tries to adjust by increasing the number of channels here to keep the signal from dying out. But, you know, best case, you're going to have slower signals. Worst case, you're going to have no signals. You know, what kind of symptoms are you going to get if you have slow or absent signals going down your axons?
It's muscle paralysis.
Yeah, you're going to have muscle weakness and paralysis on the motor side. What about on the sensory side?
Diminished or no sensory.
Yeah, people
At all.
start going. I had a student whose vision was starting to disappear, was starting to get worse and worse because of this. You know, the multiple sclerosis, young person in their 20s. You never know when it's going to start or why it's happening, but all of a sudden the vision was starting to get worse and worse because the sensory neurons were no longer sending their messages efficiently. But, you know, it also gets to be where somebody has trouble moving their body or swallowing. You can't swallow even because that's all muscles that are running. So it gives you a sense of what happens if you don't have the myelin and you don't have the ability to keep the signal high. Then the signal starts dying out. So other comments or questions?
Is the dementia or Alzheimer diseases related to the myelin?
That's something different. We'll talk about that a little later, but that's something different. There's something way more horrible that's happening with just like your neurons like dying off and everything there. So that's something more intense. Other questions? Okay, so for your exam, on Tuesday, you know, we are going to start at 8:10, so you got to make sure you're here. We are going to be using Proctorio, so you need to make sure your computer is good to go for Proctorio. If you were having issues, hopefully you've kind of figured that out at this point. I know some people were having a lot of trouble, so make sure you've got your audio, your visual, and everything all good. The material is going to be everything that we've covered up to right this moment in time. We are not, you know, I'm leaving out the actual different types of neurotransmitters. We're going to talk about that next week. Those will not be on your exam. But everything we've talked about, from basic chemistry to cellular respiration to all the different kind of, you know, transepithelial transport and membrane proteins and different kinds of potentials that we've just discussed. This is all fair game on your exam. If you have questions as you are studying, please send me emails. I'm happy to respond and kind of help clarify things. Remember, there is that study guide. It's Appendix A of your lab manual. It's also linked on the home page. That's a really useful resource to see what you're actually responsible for Your notes are important place to see what you're responsible for. So are there any questions about the actual exam that's going to happen? Lauren?
What is the format? Will there be multiple choice and short answer?
Yes.
Okay.
Yeah, so it's going to be probably like half of each. You know, half of it will be kind of short answer stuff where you have to just type in a little essay question. And half of it will be kind of multiple choice. You know, so it could be questions like, I don't know, like describe the difference between secondary versus primary active transport. And you have to tell me in your words what that means. Or it could be a multiple choice thing of like, primary active transport uses like, ATP directly, uses a concentration gradient indirectly, is a type of passive transport. Pick the correct answer. Either of those would be fair game to like ask about, active transport. So it'll be some combination of the two. Yeah, I mean, ideally it would be like all short answer, but then it becomes so hard to finish grading everything. So trying to find things that are easy to ask. But then also short answers usually are where I can really believe you understand it, because people can like, guess or kind of sort of get the right answer and pick the right multiple choice. But to actually answer something in your own words as a short answer is much more convincing to me that you really own the concept. So other questions about the exam?
So looking at the study guide, you're just saying that basically it's up until the synapse, so we're not going to have anything on functional classifications?
Yeah, no functional classifications of neurons or anything like that. Later on, we're going to be talking about afferent versus efferent versus interneurons and things, but not yet. This was more just really laying the groundwork for what is a neuron and how does it do its basic functions.
How long is the test supposed to go?
It's officially designed to be about an hour and a half, the length of the class. I usually let people go a little longer. I usually like people to, get it because they, get it because they know it or don't know it. But, especially in the world of online stuff where people start getting tempted to like look stuff up and stuff, we tend to be more careful about time limiting. So, it's the plan is to have it run for an hour and a half. If there's some reason you're having real trouble getting it done, you can like communicate with me and we can see what's going on and maybe extend things. In general, I try not to, again, have somebody miss something because they ran out of time, but they knew it. But sometimes, if you know it, typically you can get it out in time. Sometimes people just sit spinning around looking at the question, but they don't know it, so it's not going to help sitting watching it.
And then one thing about taking the test, I mean, what's different than being in person is that you can't really have like a scratch paper just to kind of like keep track of questions you've answered and write little notes down for yourself during the test. When we like kind of scan the room, could we like show you a blank piece of paper?
Yeah,
And
so that would be okay if you wanted to have like, but I need to make sure that it's not like notes ahead of time.
right. So like scan it. I don't know, like how you would want it, but so that would be an option.
That could be an option. Yeah, because I do know it kind of, scribbling stuff down is a really useful way. And normally if you have a paper test in front, you can just scribble on the piece of paper. That's your test. But
can we maybe use a whiteboard?
What do you mean a whiteboard?
Like a small whiteboard instead of a paper, and we can show you the whiteboard so you know that it's not like a notes or anything.
What do you, can you show me? Do you have it on you right now?
Yeah, it's just like a small whiteboard.
Yeah, I guess. Sure. Sure. Why not just use a piece of paper? But yeah, you can use a whiteboard, I guess. Other questions? Morgan, I just saw you wink in.
The whiteboard is a common thing in online testing, because that way you don't take notes on the test and then save it to sell on eBay later or whatever, what the test subject was.
Fascinating.